All right, now we're going to follow up on the vesicle a little bit, and we're going to be talking mostly about Chapter 6 stuff, about the cell. And so learning about the components of cell by following some dynamic features of the cell. And so we've got these vesicles inside the cell, and they're moving around, and the vesicle is composed of a phospholipid bilayer, just like the cell membrane. And so we're going to talk a little bit about it. So we've learned that the vesicles can transport newly synthesized proteins, like that, to different places. So we talked about that in class, a previous video. And then here's another thing that that vesicles can transport. So here's the phospholipid membrane of this vesicle. So when we draw this as a circle, it's got a phospholipid membrane, and we've learned that it could fuse with the cell membrane, and it could add its phospholipids to the cell membrane. And that happens because where do these phospholipids get synthesized? And the answer is that they typically get synthesized by the smooth ER, the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum is doing something very different than what we learned the rough ER is doing. The rough ER is an important site of protein synthesis. So it's one of the important places where proteins are getting made. What's happening at the smooth ER is that this is a place where hydrophobic molecules get metabolized. And when we say metabolized, we'll learn in Chapter 9, in time for our midterm, we'll learn that sometimes metabolism involves breaking things down, digesting things. That's catabolism. Or sometimes it involves building things up. And both of those happen in the smooth ER. The smooth ER has lots of membranes because it's in the membranes of the smooth ER where this metabolism occurring, because the membrane is hydrophobic, and so we can have these hydrophobic molecules getting synthesized there. What are some examples of the synthesis? Well, phospholipids can get synthesized there, and then they can get delivered through vesicles to the various destinations that are made up of phospholipids, like the cell membrane itself. And so newly synthesized phospholipids come from the smooth ER. And lots of other molecules too, but that's our example. Newly synthesized phospholipids come from the smooth ER. They got to be delivered by these vesicles because they have these hydrophobic tails, and we're in a hydrophilic water environment. So, it's a key part of this is that these hydrophobic molecules need to be held in a specific location and moved through using the hydrophobic parts of the cell of the membrane of the vesicle. So not only do newly synthesized proteins get moved around by vesicles, but there is transport of hydrophobic molecules in the membrane of the vesicles, and that includes newly synthesized phospholipids and lots of other molecules too. Okay, so the next thing we're going to do is we're going to talk about a couple other examples of moving vesicles around. And there's a figure. Oh, here's one from class. So in class, I brought up it's just what's the purpose here is it's just a quick example of another thing that gets moved in a vesicle. And so this is a Wikipedia page about this medicine called Botox that helps relax muscles. And I talked about how it helps relax muscles. What Botox does is it blocks the release of a neurotransmitter called acetylcholine. And acetylcholine is carried in a vesicle, stored in a vesicle, and when it's time to stimulate muscle contraction, this is a muscle cell here, this is a nerve cell here, and the nerve cell will stimulate muscle contraction by releasing acetylcholine, and it occurs by fusing this through the vesicle and exocytosis, releasing acetylcholine into the extracellular environment, and it binds to receptors on the muscles and causes muscle contraction. And Botox blocks the fusion by blocking important proteins that help this vesicle dock. So the bottom line here is here's another example of materials moved in vesicles, and it is in vesicles. So we got newly synthesized protein, we got newly synthesized phospholipids, we've got neurotransmitters. So in our nervous system, this is happening all the time. Anytime you're thinking, you're releasing neurotransmitters. And in class, we talked a little bit about drug addiction and how drug addiction affects both legal and illegal drugs can affect this release of neurotransmitter by vesicles. So we're going to have a couple more examples of things moving in vesicles. One of them is going to be phagocytosis from your book, and one of them is going to be called receptor-mediated endocytosis. So we'll deal with phagocytosis, and then we'll talk about receptor-mediated endocytosis in the next video. Okay, so here is a figure from your book where those are dealt with, and here's phagocytosis, and the example is that we're going to focus on is going to be there are cells in our blood. There are red blood cells carrying oxygen. There are white blood cells that are involved in our immune system, and we're going to focus on an example of a white blood cell that's called a macrophage. We talked about that cell a couple of times. And macrophages help kill bacteria that are causing an infection. And here's how they do it. The macrophage finds the bacteria, and the bacteria, here's the bacteria, gets surrounded by the membrane of the macrophage and then brought into the cell by a process called phagocytosis, which is one type of endocytosis, bringing stuff in, endocytosis. And here's the bacteria now inside the cell, inside a vesicle. And that bacteria is alive, it's causing an infection in your body, and then that vesicle fuses with a lysosome, and the acidic lumen of the lysosome wipes out that bacteria. It causes the bacteria, it kills the bacteria and digests it, and the bacteria's components like its amino acids are recycled inside the macrophage. One of the details of this is here's your blood vessel. Often these living bacteria that are causing an infection are outside of the blood in your tissue, and the macrophages need to leave the blood, crawl between the cells and the wall of your blood vessels, and find the bacteria and phagocytose them. And you'll see in two videos we're going to talk about the cell movement, another part of chapter 6, so this dynamic cell, and we'll talk about a macrophage crawling out to go after these guys and phagocytose them. Then here's just another example of phagocytosis in your book, cell drinking, where a vesicle is created that is just pulling in contents outside of the cell and bringing them into the cell like that. And then in our next video we're going to talk about this, a final example of vesicle traffic, and it's receptor-mediated endocytosis. And so we'll talk about that in our next video.